All right. So, we'll just get started. So I'll make an introduction. So it's our great pleasure today and to have Dr. Bozhi Tian from University of Chicago. Sorry. University of Chicago visiting us. I didn't know Bozhi before. We never met before, but of course I know his work. And Bozhi got trained from Harvard University and with Charles Lieber, and many of you heard of him. So then he went to MIT for postdoc training and with Dr. Bob Langer and in tissue engineering. And afterwards, he went to University of Chicago, and now he's a full professor there and establishing a very, very productive, prominent group working on areas using bioelectronics, semiconductor-based approaches to study, understanding, biophysical aspects of living systems, and also studying questions and considering, the interface between soft and hard materials. And recognizing his research excellence and innovation impact, he has received many, many different awards, and he's so moderate. And the bio he sent to us, I only saw one award, but of course, I assume that's the award he cherished most. Is PK's award. Is what we call Presidential Early Career Award for Scientists and Engineers. And this is considered as the best award for engineers. But to be frank, he has many, many other awards worth mentioning, including NIH New Innovator Award. And of course, as well as NSF Career Award. As long as you have to check that box for engineers. Okay, without further ado, one thing I want to mention, I met Bozhi this morning for half an hour, and at the end, one thing I want to mention, he told me he believed he's engineer in Harvard. But nonetheless, I think sitting with his lab in chemistry department and laying, with the solid foundation in chemistry and material science, I think those solid foundations allow him to, pursue some very important engineering applications. So without further ado,
well, thank you. Thank you very much. Thank you very much for the very nice introduction, Professor Fu. Good afternoon, everyone. My name is Bozhi Tian. I'm a material scientist at the University of Chicago. In my lab, we have been working on several different areas. For example, we're excited about what we call living bioelectronics, where we blur the distinction between living and non-living systems. We're also interested in regenerative medicine, where we have designed a few ways to heal the tissue. It can be engineered tissue, it can be a real tissue. But the most important area that we have been working for almost a decade, that is the photostimulation of cells and tissues, or you can more broadly say this is the bioelectronic modulation area, and you will see a lot of details here. At the heart of our research are the deformable materials that we have designed, characterized, and also applied in a range of biomedical applications. And our work spans several types, the materials span several types, including the delicate layers of semiconductors and adaptable, I see, sorry for that. Okay. All right, let me, okay. Including the delicate layer of semiconductors to adaptable granular composites and all the way to the study of minuscule microbes. And today you will see how we utilize those different material building blocks for a range of studies. Apologize for the connection issue. And today I will present mostly three different stories, but they are all connected by this deformable composite, that central theme. First, I will tell you a little bit about the photostimulation of cells and tissue. As I said, this has been, we have been working on this topic for almost a decade. But today I will only focus on the translational part. You will see the latest ones, how we utilize this material and this technology and push this as almost a translational cardiac pacemaker. Not really there yet, but it's very close. I will also tell you some story about granular materials, and specifically granular materials-based hydrogel composite for regenerative medicine and bioelectronic recording. And finally, I will introduce the latest work, still unpublished, is living bioelectronics. So living bioelectronics means that we have to incorporate some living cells. And the living cells here are bacteria cells, but they're good bacteria. They're commensal bacteria that can secrete some very useful pharmaceutical agents that can help treatment such as the psoriasis. So you'll see a little bit details later. This is my very first time at the University of Michigan. So I do want to take this opportunity to highlight some other pieces of works that might be relevant to mechanical engineering, but may not fit into the central theme I'm going to convey today. For example, our lab is interested in sustainability. And one of my postdocs, Chuan Yang, he has been working on large-scale fabrication and specifically roll-to-roll fabrication of electronics and robotics and using a greener approach, sustainable method. So we use water, for example, instead of toxic chemicals for isolation of devices and mass production of devices. One of my former graduates in Lingyan, she's now in industry, and one of her last PhD works is about the engineering of a what we call hexapod, but use this hexapod for T-cell activation and signaling study. Specifically, this hexapod contains a magnetic core and insulating silicon dioxide branch, and under a rotating magnetic field, those rotational motion of the hexapod can interact with T-cell and enhance the T-cell signaling process. But another work that I really would like to highlight today is the work done led by Jincheng Ma. So he got a training at a UIUC mechanical engineering department. And during his stay, less than 2 years stay in my lab, he has brought up an ingenious design for gas sensing. And this is a portable device. And this device can allow the detection directly from the vapor phase, a range of chemicals. And the basic idea here is to utilize this, what we call multi-phase condensation. So by the way, he was trained in the field of thermofluidics. So he utilized his expertise and integrated with our bioelectronics area. So what we can do is to directly condense some gas vapors from ambient and form those condensates. Sorry about this. Let me see if this connection can be better. And using this approach, we can do environment and food safety and also even the healthcare monitoring. And through this method, we can also significantly improve the sensitivity by 7 orders of magnitude. So it's a big improvement. And I will not show you all the examples, but just to let you know that we can actually detect glucose from exhaled breath and in a non-contact mode. It's just to give you an idea how our other areas are going right now. As I promised earlier, I will focus on the deformable composites, and we'll start with the first story, the photostimulation cells and tissues. I'm a material scientist. The very first step for material scientists in any type of research probably is to design and synthesize new materials or fabricate a material-based device. In my early career, I focused on neuromodulation, and at that time, I was interested in central nervous system. And there are a lot of materials that can be interfaced with brain. For example, the upconversion nanoparticles, magnetic particles, you can use them for either deep brain neuromodulation or for imaging purpose. Our lab has been focused on semiconductor-based systems, and we use semiconductors because they can be configured into a range of important device configurations, like FET transistor or a photovoltaic device. When I was a graduate student, I used FET transistor or FET for electrochemical for electronic sensing from cells and tissues. And after I became independent, I used photoelectrochemical effect for modulation. And I will come back to some basics in a minute. Those two diagrams just want to give you a very general, broad overview of the photoelectrochemical process under physiological condition. The plot on the left side shows you the band-edge positions of a few biocompatible semiconductors with respect to the potential for vacuum potential and also normal hydrogen electric potential. So based on this diagram, you can almost predict what would happen at the surface of semiconductors under either electrochemical condition or full electrochemical condition. The plot shown on the right side shows that there's a band bending at the semiconductor and the silicon interface. Upon light illumination, there could be some photocarrier generation. Some of the carriers, let's say electrons or holes, can get accumulated at the interface between the semiconductor and silicon. then some of the carriers can introduce at least a transient change in either the concentration or identity of the chemical species, and that could be felt by cells and tissues. So then cells feel this, or tissue feel this, they can have some changes in their either electrophysiology activities or other processes. This underlie the biological modulation using a photoelectrochemical effect. So it's not that complicated at all. So think this way, think about a solar cell, sunlight produce some electron output. That electron output can cause some transient or even permanent change in chemical species. And the cells and tissue can feel that and got excited or inhibited. So just that simple. Yuanwen and Ramya, they were the students I recruited in my first year. And I'm very proud of them. And Yuanwen now is assistant professor at UPenn Material Science and Engineering Department. They took slightly different methods for biological modulation. Ramya used the bottom-up method. Basically, we start with the synthesis of nanostructured building blocks, like silicon nanowires. Then we can also assemble them into a microscopic sheet supported by some polymer mesh. You can either form a single nanowire and a single neuron junction, or a single tissue or single organ and a microscopic sheet junction using light for training or stimulation. Raman used top-down method, so he can use commercially available silicon wafer like SOI wafer, using lithography, make them like island-like a thin membrane, and also laminated with some PDMS membrane. The PDMS can contain a lot of holes, and then you can laminate them together, form a deformable composite, and put on the surface of the cortex. This is the anesthetized mouse. So by itself, it will not elicit this reaction. But then you shine light through this transducer, you can call it the transducer, you can elicit some limb motion. So this just gives you a very general idea how this neuromodulation or in general biological modulation would work. Now, as a scientist, a very important thing is to understand the mechanism. In our lab, we use two-step method for understanding this mechanism. What the mechanism? Modulation mechanism. The first step involves materials only. No cells, no tissues. We use a setup, essentially it's the modified patch clamp setup. So we have a glass pipette, we have our silicon materials or other semiconductors, then shine light, then use pipette to measure the photoelectrochemical current, not photoelectrochemical, the photocurrent from the surface of the silicon materials. Now, the key thing is that the photocurrent contains two components, thermal component and electric component. What does that mean? That means when you shine light to a material, the two most dominant effects from a semiconductor material would be either photoelectrochemical output or photothermal output under physiological conditions. That's as expected, right? And two effects, thermal or electric, either can stimulate neurons. So for us, we actually need to understand which one is dominant, which one is minor. How can we do this? The good news is that those two components, they have different dependence on the pipette holding potential. So what we could do is to change the pipette holding potential. At each potential, we measure the photocurrent, then we can decouple them just using a very simple algorithm, nothing special. After we decouple those two, you would know which one is bigger, which one is smaller, or which one is dominant, which one is minor. This is just the first evaluation. After the first step, we have a rough idea if this modulation is going through for the electrochemical or for the thermal. The second step is to build an actual biointerface. In this case, we have a neuromodulation interface between a single silicon wire and a single neuron. Then we can shine light either by changing the duration or changing the power. You can have a switch from a subthreshold depolarization to a full action potential. By the way, the output was measured using patch clamp, which is the gold standard for intracellular electrophysiology. After you have this sudden switch, you know that there's a threshold. If you plot this power threshold against the power duration, you get the so-called optical excitability curve. Now, here's the key. For two mechanisms, photothermal or photoelectrochemical, so given time, I won't go into the details. The shape will be different, the exponent, right? So you can see there's a difference. For this specific case, it follows this one, suggesting that for this biointerface, the modulation follows the photoelectrochemical effect instead of photothermal effect. Through those two-step evaluations, we will have a much better understanding of the modulation mechanism, and it is important for us. Throughout the years, our lab has used light as the principal input, semiconductor as the transducer. we have achieved inter, and extracellular modulations in the nervous system, cardiovascular system, and even a microbial system. But also would like to highlight a few other pieces of work from other research labs across the globe. And in particular, I actually want to highlight Professor Jun Tang's work a few years ago. And this is really one of the highlights I would like to put here. Just want to let you know that this is a very good community. We support each other and we're trying to do better work together. Before I move on, I just want to say a few things about the key differences between our approach and the two dominant methods currently practiced in the field. Okay. The first one is electro-based method. And this has been practiced for centuries. If you think about the cardiac pacemaker, the current cardiac pacemaker, or the deep brain stimulator, they are essentially electro-based method, right? And the good thing about this is in terms of the translational application is certainly great because it's non-genetic. You can either use this for patients without too much trouble if it's FDA approved. But in terms of operation, it is not flexible. What does that mean? Upon implantation, the location for sensory stimulation is really limited by where you implant your electrodes. You cannot arbitrarily change the electrode location unless you do a secondary surgery. The second method is called optogenetic, has been extremely powerful since 2005. It has reviewed a lot of underlying neurological disease mechanism. It uses light, so in principle, you can achieve random access. Random access means if you switch a light spot from here, spot A to spot B, you can either change the location for sensing or stimulation. It's just that easy. However, if you look at the name, right, it involves genetic modification. It works perfectly for small rodent models or in vitro culture. But if you want to work this method in the larger brain mammals, not to mention human subject, there will be both ethical and technical hurdle. So ethical hurdle is obvious. The technical hurdle is the off-target effect, just one of them. Our method involves the light, so it combines the advantage of this, but also it's non-genetic, although it uses semiconductor as a transducer. So there are some advantages of using this, but there are currently there is still the limitation. One big limitation is we cannot achieve cell or circuit level targeting. Now let me tell you some examples so that you can understand better our approach. This work was published about 2 years ago, done by my former very talented graduate student, Alex Perminsky, and my former postdoc, now assistant professor at Technion, Amy Rutenberg. I won't go into the backstory, but it was a serendipitous discovery. It was not made by design. By luck, we found some really good material. And I would directly go to the conclusion. The conclusion is that we discovered by accident a heterojunction we call porosity-based heterojunction. It contains two layers. One layer is non-porous second layer. Essentially, it's the commercially available silicon wafer. And another layer is nanoporous layer. It can be 1 micron to 5 micrometers with a pore size on the order of a few nanometers. How can we generate nanoporous layer? By thin etching, which is also decades old method, but we found this by accident. Now, the final outcome is the material gives a really good photoelectrochemical output. It's so good that it's actually the best material that we developed in the lab so far. We use for a range of modulation, neuromodulation, and cardiac modulation. Now, let's look at the structure. This is a cross-section scanning transmission electron microscopy image showing the non-porous part, the interface, and nanoporous part. These electron diffraction patterns recorded from the 3 different areas: non-porous part, interface, and nanoporous part. What you can see is that those 3 patterns actually are almost the same, but they're slightly different, suggesting it's a coherent single crystal throughout the entire material. If you zoom in, this nanoporous region, the feature size or the wall thickness is about a couple of nanometers. When silicon becomes this small, the quantum confinement effect kicks in, and you would expect to have a larger band gap for silicon, although you start with the same silicon, non-porous, but after etching become nanoporous structure, the band structure changes. The Fermi level position with respect to conduction and valence band will also change. When reaching equilibrium, those 2 layers, non-porous and nanoporous part, they form what we call a staggered type 2 heterojunction, which is quite similar to traditional p-n junction, which is the building block for solar cell structure. So now you can see, comparing those two structures, both can have photovoltaic or photoelectrochemical effect, although we do not need to involve N-type doping. And the whole processing is significantly easier. We do not need to go to clean room or very sophisticated environment for doping or for microfabrication. The etching takes 10 seconds, no more than 1 minute. You can achieve this, and you just use commercially available silicon wafer. Now let me show you just a video. This video was pretty old, but at least it can give you a general idea about how photostimulation cells and tissue work. This is the isolated heart from a rat model. The dimension is about 1 to 2 centimeters, depending on the age of the rat. On the surface of the heart, you see a piece of silicon membrane, but it's not a monolithic. There are 7 domains. This is the commercial. This is just a traditional electrode we use here as ECG recording. And this is light source. We get light pulses, in this case, 2 hertz. Piece of paper initially blocks this light, so the light will not hit the heart surface. But after we remove the paper, as you can see from this, right, initially slow, but then after you remove paper, the light hit the surface, the heart beat much more quickly, got synchronized to the light frequency, which is 2 hertz. This is called optical overdrive pacing, meaning that 1 light pulse, 1 pacing. And you should not miss a pacing. If you miss it, you cannot call overdrive pacing. Okay, if you put the paper back, the heart beating slow down. For many years, we have, sorry.
Where was the electrode placed exactly?
You're talking about this electrode, or? So there's no electrode here.
Polysilicon structure.
So the silicon is placed here.
Yeah.
Yeah, there is.
But on a cellular level, relative to the, let's say, anatomy of the heart, where was it placed?
So I want to make sure you're answering your question correct. So you're talking about
I'm talking.
left ventricle or right ventricle, right?
Trying
Yeah.
to understand the physiological
Right.
mechanism, right?
I see. Yeah.
You excite some cells.
Yeah.
And so I'm asking which cells
Right.
in the heart are you exciting, and why does it happen to bring about this accelerated contraction?
Right. In this case, left ventricle, but we can actually switch to right ventricle as well. And you can do stimulation in this either case.
It doesn't, it's not interfacing with the electrical conduction system.
Well, you will see later on. Yeah. Well, you'll see more precise full stimulation later. Yeah, but a good question. So we have to have that precision exactly as you said. And this is what this paper is trying to address. For many years, we have been thinking, how can we improve the precision of stimulation, right? It's not just to one spot, but to multiple locations so that you can actually target a certain like conduction pathway. And the question is like this, but it's hard to address. Why? If you use just monolithic silicon membrane, if you think about traditional solar cell, when you shine light, that photogenic carriers can spread to a very large area. And this is very good for traditional solar cell because this is good for carrier collection. You want something like this, but it's very bad for photostimulation because if it diffuse, you cannot differentiate those 2 light spot location, the impact from this light spot location because the carriers essentially got blurred. What we hope to achieve is a highly localized photostimulation. You have light spot shining here. You only generate photostimulation from this location. If you switch to different location, you produce a different photostimulation impact. If you think about our biology, there's a lot of locations you can think about random access. Right? Retina, you shine light to different locations, different parts of the receptors can register that, although you don't need a pixel at all. And even for our skin, using mechanical receptors, you can easily identify the location where you press your finger or have some pressure, external pressure. So you need that type of random access or pixelless capability in a synthetic modulator or a biological modulation tool. So how can we tackle that? So this is probably one of my best graduates in Punjuli, and he recently really made this happen. I won't go into the specific details, but he has screened multiple silicon-based hydrojunctions, including the commercially available single crystalline p-n junctions. The different color represent different doping type, p-type and n-type. Then pin silicon, so p-type silicon, nanocrystalline intrinsic layer, and nanocrystalline n-type layer. So it's pin. And then pin, but decorated with the gold nanoparticle on the top surface of the n-type nanocrystalline layer. And finally, this nanoporous silicon that I mentioned earlier today. So for the first three materials, the photogenetic electrons, they're majority carriers in n-type layers, so they can traverse a larger distance. However, for the last one, ever since the p-type, the photogenetic electrons are minority carriers, so they can their travel distance will be highly confined. But there's also other nanostructural aspect that makes this one potentially the best localization material. We also studied the impact of the geometry, for example, the scale-like or Kirigami-like geometry, how that impact the localization. Given time, I want to focus on that. But just want to show you some photocurrent mapping result. In those mapping results that we collected from the 4 different materials, we shine light to the lower left corner of the silicon wafer. So silicon wafer dimension is about 4 millimeter by 4 millimeter square like. Shine light here. And what do you see here? The different color represent different polarity of the photoelectrochemical current. Red means it's a cathodic current, photocathodic current, and blue means photoanodic current. If you see a boundary or even boundary evolution, that means there's a transition from one polarity to a different polarity. So the first takeaway message is that in terms of localization, this material is the best, as predicted by the minority carrier argument I made just now. But additionally, you see that 2 polarities can show up for those 3 materials, but for this traditional p-n junction, there's only 1 polarity. This charge balancing is actually important for a lot of biointerface material because you don't want to locally accumulate a lot of toxic species. You have the charge balance is actually better in terms of biocompatibility. So 2 messages: 1, localization is the better, and second, in terms of the charge balancing, you have 2 polarized domains on the same surface, and the center is only defined by the light spot. After showing this, we first demonstrate the random access photostimulation in a vitro system. This is the cultured submonomer of cardiomyocytes. Using this setup, so you shine light from bottom, and there's a cultured cardiomyocyte, and the silicon chip is flipped, right? So then you shine light and produce localized photoelectrochemical effect. And we want to see if that can produce a random access photostimulation. For example, you can change the light spot size so that you can control many cells or just down to single 1 or 2 cells. You can actually achieve subcellular manipulation as well. Additionally, we can change the location. For example, you start from here stimulation, start here or here, and your initiation point, bioelectric initiation in that cardiomyocyte submonomer is also different. You can use either membrane voltage dye or calcium imaging dye to verify that. This is just the in vitro verification, but we have to go to the tissue, see how that works. This is still a overdrive pacing video, except that now you see 6 Hz pacing, so it's probably more efficient. Indeed, the power we use here is only a fraction of the sunlight intensity. Some of you are working with solar cell using 1 similar light intensity. This is just a fraction of that. And you can do that for the stimulation. This is not random access because the light is always hit in the center, close to the center of the silicon membrane. This can address the random access, or at least close to that. This is a silicon membrane, but the center is open, where we put a microelectrode array for cardiac mapping. So there are both stimulation component and sensing component. You can do photostimulation at different spots, like P1, P2, P3, all the way to P8. What you found from this mapping is that there is the cardiac mapping, the isochrome mapping. You can see that when you hit light to different locations, it actually gives you a different result, suggesting that there's a need for those random access photostimulation or precision photostimulation. Now we can even scale this up, right? So now you have a membrane, monolithic membrane placed here, The dots, they're artificial, they're not real dots, this is label location. But now you can stimulate up to 100 different sites, place, the membrane is placed left ventricle and between left and right ventricle. And then you can see that when you hit the light, different location, there is a slight variation in the recorded ECG signal. This is, by the way, the in vivo in the red heart configuration. The previous one, that was the ex vivo, that's the isolated heart. For many years, when I went out and gave lecture about, give seminars about photostimulation, one of the most frequent questions I received from the audience was, well, your method is non-genetic, but so far, all the animal models you showed us, they're rodent models, and optogenetics can work with rodent model. So what's the home run? What's the killer experiments that can change the game? So we decided to move on to this pig model because this is something that optogenetic can not work with at current stage. So again, Peng and together with our collaborator made this happen. This is the open chest surgery. This is the pig heart. The pig heart is even bigger than the human heart. The pig is more than 200 pounds. On the surface, there is a piece of a silicon membrane. It's a monolithic membrane without islands, without pixels. And this is optical fiber. This is just a very simple experiment demonstrating showing that overdrive optical pacing in the larger animal is possible. Here it shows the data trace before and after. You can see a increase of the beating rate up to the 2 hertz. But you can still say this is too invasive, and I agree with you. This is the invasive. A medical doctor won't adopt this. So how can we further improve this? My student Pengju has designed and also engineered a minimally invasive tool. This essentially is catheter-like tubing, and using this tubing, you can deliver, unfold, and attach silicon membrane on the heart surface. And through the same tubing, you can deliver optical fiber. So on the next slide, you will see how that works. This is the device that he made using a 3D printer and delivered through, this is the side view, this is the top-down view. And you can put this membrane on the heart surface. This is the sheep dabber model. And after that, this is on the real heart pig. Then you deliver optical fiber and give light pulses, and you can achieve overdrive pacing. So at this stage, this is still very preliminary, and we're having some ongoing effort right now, still trying to make the cardiac pacemaker better, and the surgery process much less invasive. So we're still working on that. So any questions so far before I move on? So this is the first part of my story, but the second and the third will be much brief. Yeah.
Does this produce a mechanically productive contraction of the heart?
Mechanical product. Sorry.
So I mean,
Yeah.
the purpose of the heart is
Right.
to
Yeah.
eject a bolus of blood.
Right.
I can send a heart into defibrillation.
Yeah.
And
So-
there is something moving,
Mhm.
but there is no blood flowing.
Mhm.
So I'm asking, is the stimulation leading to a productive cardiac cycle mechanically?
Yeah, so this is a great question. We haven't investigated that part yet. So our initial effort is to see whether the photostimulation in terms of the bioelectric activity, if that first step can be accomplished.
I
Yeah.
don't know this, but
Yeah.
in pig physiology,
Mm.
has anybody ever taken the equivalent of an electrocardiogram?
Electrocardiogram on a pig model.
You know, if I would, oh, my chest
Yeah.
hurts, and
Well, I'm sure someone has collected this. Yeah, I haven't checked it.
emergency room,
Yeah.
they take an ECG, and they would know exactly within minutes
Mhm.
if my heart is properly electrically activated.
Mhm.
So is that technique available for pigs? Can you do
Well,
the
I think people who
equivalent?
are doing that probably is, yeah.
In the simplest case, you just need 3 electrodes,
Mhm.
one arm, one leg.
Right, so you can certainly detect that. That's the sensing component.
Correct or incorrect spatiotemporal innervation of the beating of the heart.
Yeah, well, I think in principle, that device could be implemented on a pig model. And that's a sensing device, so you can monitor that. You see the problems, put on pop. But if you want to correct that, you still need some device. And for that device, if you want to go to a temporary pacemaker, because we're not trying to compete with permanent pacemaker, we're trying to develop a niche for temporary or transient cardiac pacemaker, and also using light, which could be less invasive or less bulkier. Our silicon membrane is just 5 micrometer, and the weight is much lighter, and it wouldn't cause any mechanical issue to the heart itself because it's so light. And the light optical fiber tip is centimeter away from the heart surface. It will not cause constraint like a traditional electrical device. Yeah, maybe I can just answer it, but we haven't moved on to the next stage, as you said, how electrical and mechanical are coupled. Yeah, we haven't got to that stage yet. Maybe the second stage. Yeah.
Also, I saw that another cool experiment could be done. Maybe
Yeah.
cardiac arrest model animal
I see.
could be available.
Yeah.
And of course, there are fundamental questions related to, say, cardiac conduction whatsoever. But I guess for those type of translation, I guess you just want the heart to stop beating,
Right,
right?
exactly.
To rescue
Yeah.
such clinical emergency as soon as possible.
Yeah, exactly. Yeah. Fast action and acute surgery. Yeah, things like this. Yeah, question.
I'm curious about the fabrication of the porous silicon.
Right.
You just do etching?
Yeah, just simple etching. Essentially a conversion of the single crystalline silicon into nanoporous silicon.
But how can you fabricate into nanometer scale? Just wet etching?
Just wet etching. We do not need template.
So will the surface, will the topology be amorphous or homogeneous?
It is a coherent single crystal. We use the electron diffraction pattern to see that. Yeah. So it's still single crystalline after etching.
I have another question is that I don't know why I'm understanding this, but in the first part of what you said, I think simply said is you shine laser onto that membranes and you can generate photo current and you can enhance the beating of heart.
Right. Well, in our case, yes, increase the beating rate.
Oh,
Yeah, of course. Okay, so now let me move on. Just talk about the second work very briefly, and this is about granular materials, granular hydrogels, and the idea is very simple. We started with a granular hydrogel. You have a hydrogel matrix. You have embedded granular particles. The granular particles are starch granules, and hydrogel mostly gelatin. Gelatin is sensitive to temperature and also pH. Upon environmental change, we can pH change, body temperature change, or even enzymatic degradation. You can remove part of or melt the hydrogel matrix and release in these granular particles. Now, these granular particle surfaces are modified so that they can attach to the tissue surfaces upon release. Now you switch from monolithic interface to focal interface. You may ask why we want to start with this, because it's easier for handling, mechanical handling. It's just about that simple. Injection or application like a patch, but later on, you may not need that large surface area for that bio interface. You need probably a smaller one, so that diffusion or other things wouldn't become an issue. This granular particle can also degrade. Eventually, it can be disappeared completely, and degradation product can be beneficial too. So there are a few scientific aspect I will show you later. It's from surface chemistry. But let me first tell you how we engineer the device. You can have a bandage structure. You can integrate it with bioelectronics, with the flexible bioelectronics, and integrate them together. Apply, I say, on the skin surface, the heart surface for recording or stimulation. You can also do rheology analysis to understand how the granules is going to get released, either by temperature increase or by pH variation. So the idea here is to look at the viscoelasticity properties, and specifically, for example, the storage modules. If your storage modules that goes down, and if you see a viscoelastic property increase or become more viscous, then the granular particle can release better. Otherwise, it will just embed it in the matrix, will never come out. You want them to come out in order to attach to the tissue surface and form a focal adhesion. So this slide is the key. How can we form a focal adhesion? And the idea here is to do surface modification Our granule particle essentially is the starch core and the catalyst shell particle. But over the catalyst shell, we also modify the material with aspirin. Aspirin is a drug, but we're not using this for therapeutic purpose. We're using this for bioaffinity. In fact, we can use a range of drugs, not just limited to aspirin. There's drug molecules, they have a very good the bioaffinity. Let me see what happens. Probably like so.
Or interface.
Sorry about this. Let's see what happens.
Yeah, internet access.
What?
Yeah, internet access right now.
Just back screen.
Yeah, try the AirPlay.
Yeah, that's a good solution. Maybe it's an airplane.
You have internet access.
No, I don't have internet access.
But I can get you connected. Then we can use maybe airplane.
You can use the EDU room, right? EDU room.
Yeah, that's what I'm saying.
Let me turn on this. Just I connect to the power. Unless
Have you seen this before?
I think it's just power off. But this one should be connected, right? This should be there should be a power. Right, it's always connected. So this one.
Let's not be in the back.
Give me a minute. Let me see how I can solve this problem. Sorry about this. Yeah, I think the battery went out.
It's charging for.
Unless this one doesn't work.
Yes.
Okay.
Why does that make a difference? The pictures, the little screen.
Okay. This one probably. Yeah, we should. Okay. Yeah. That.
Much more.
Also the same. Right, thermal can also work. Yeah, and of course it's thermal is very, very straightforward. Yeah. Stimulate your semiconductor devices. Okay. Whether it's a cell phone, it's
Physical logic.
Let's get sketchy.
In the answers we don't know.
It's all right.
Whether this.
Okay, just check whether it's charging or not.
Oh,
Blood
Yeah.
pressure. So in
Okay.
million ways of.
Just make sure it's charging.
More like in the downstream, that may not be that.
It is charging. Okay, so it is charging.
Provided. Especially if you are measuring like somewhere, like further down.
Well, if it's charged, yeah, then this one should work.
That's why I would suggest FM.
Be close to the heart. And you should see a nice thing happen. Of course, if you stimulate it at a frequency that represents fibrillation, we know that it's not functional. That's an entire point about defibrillation being an cardiac procedure.
应该是有的，应该是有的，应该是有的。
Very.
I think we're doing.
There's that guy.
We're down to a chalk.
Sorry about this. Yeah.
It's okay.
Sorry about that.
My office.
I could see that majority of it. Yeah, I actually finished majority of this. Yeah. We already lost for like 30 minutes. So the other things are just relatively better.
Yeah.
The last science paper is not full there. It's not online yet.
Yeah, it will be online in about 1 month. Yeah, about 1 month. Yeah.
I think it's like all the exciting work. Stuff
Yeah.
It takes some time.
Yeah, it takes some time. So, I'm doing some temporary charging. Right now, I cannot open my laptop. It will take just some time just to do some temporary charging. And I'm open to answer any of your questions. Maybe I can also just verbally say, I only have a few minutes left, right, slides. And for that granular thing, the monolithic to focal biointerface, that transition thing, the application we're thinking about, we actually demonstrated the treatment of the using the Kaleidos model. We also have used that device put on the hard surface, and we see an enhanced bioelectronic recording because that focal adhesion that occupy much less surface area. I also mentioned that surface modification aspirin. So the aspirin together with other drug molecule, as mentioned, that we're not using this for therapeutic purpose, but the drug molecules, they can interface with the cell membrane very strongly. Otherwise, we will not take that drug. We use that bioaffinity as a mechanism for focal adhesion. So essentially modify surface with drug molecule so that that granular particles can attach to the tissue surface much better. So that's the basic idea. Essentially use drug molecule as a glue. So application treatment of the Kaleidos model and the hard recording. The last story that I unfortunately don't have time to mention is about living bioelectronics. So my lab has been actually, when I was a postdoc, we did a work we called cyborg tissue. Basic idea is that we have engineered tissue, and we incorporate some bioelectronic, flexible bioelectronics inside the three-dimensional engineered tissue. At that time, we use this to test the drug effect. We do a lot of screening, and we measure the cardiac activities from the engineered tissue. So back to that time, that was the goal. But later on, I realized that it's still far away from translational applications. If you want to move to in vivo studies, you have to solve a lot of problems such as the biocompatibility issue and other things. So over the years, in my lab, we have been thinking about solutions to that. And only until recently, we finally solved this issue. So we have a living bioelectronics and can be used for in vivo application. And at this stage, it's still attached to the skin. So we're using this living bioelectronic device for treating psoriasis. So kind of a skin inflammation. And the basic idea is that we incorporate some commensal bacteria. It's called SE6. And the SE6 can secrete molecules that can treat that inflammation. We use bioelectronic components to stimulate bacteria, enhance their activities, or we can inhibit the activities. So at the end of the lifetime of the device, you can pass a large current and kill all the bacteria so that you don't have any infection problem. But when the bacteria are still active, you can use electric current to modulate the activity and produce therapeutic molecules. The bioelectronic components also contain sensors that we can use for temperature measurement and humidity measurement, and also electric impedance. And that could be used to monitor the inflammation progression and also the recovery process. So that's basically the story. My talk today. Let me see if I can turn on my computer now.
Open up for discussion, I guess.
Yeah, feel free to ask me questions. I apologize for this issue.
There's some, maybe you're first. Go ahead.
So essentially a question about first one, maybe I've missed it.
Yeah.
Well, since the heart is a constantly beating structure, so I assume there will be non-trivial mechanical deformation inflicted in your devices, right? So do you incorporate any microstructure to accommodate the deformation, or there are some kind of tricks you employ to address that?
This is a very good question. So your question is, in our monolithic membrane, let's say if there's deformation, it might crack or other things or damage. We have holes, periodic holes, that we inserted into the membrane. That holes were generated by reactive ion etching. That holes can essentially deflect the crack propagation. So even there's a deformation, the membrane can still survive. And especially our membrane is only 1 to 2 micrometers. Yeah. So without thickness, it should be fine. Yeah. Yeah. Thank you.
Very nice thoughts.
Thank you. I apologize for the issue with the computer.
No, first
Yeah.
time learning about this, it's fantastic. So I want to go back to your kind of when you compare the three approaches, the electrode, the optogenetics, and kind of the semiconductors. Kind of now that we've seen this, can you comment on like what are some of the problems where each of these approaches are sort of best suited for tackling?
The problem with our current method, right? Yeah.
Like what type of what problems do each of these address kind of?
The problem, okay, yeah,
What is
absolutely.
best suited for addressing?
Yeah, applications, yes. For example, the traditional electrode-based method, this has been practiced for centuries and used for clinical applications. And people, I would say there are, you didn't like deep, you think about cardiac pacemaker, deep brain stimulator, right? So there are a lot of diseases that you can target. And essentially most of the current implant devices are based bioelectronic device for therapy, essentially based on electrodes. All the problems are addressed by that, but that's the current thing. Now, optogenetics, I think mostly for fundamental studies, at this stage, for clinical application, there is still, people are trying to use some simple model like retina or bladder. Those probably can work with optogenetics. But for other part of the body, there's still a way to go. Yeah.
I see.
For our method, we hope to do translation studies. Yeah.
So I think the idea is the body is very sensitive to electrical stimulation.
Right.
It's not very sensitive to optical stimulation. You really have to try.
Well, very few places can saturate the light.
I guess.
Yeah.
Right? So for most of our body, yes.
That's why people use optogenetics, right?
I see.
Engineer cells, tissues, so that they can respond to light.
Right. But then you need to engineer the cells, right?
Right,
I think
right.
that's- Okay.
That add a lot of complexity.
Thank you.
Yeah, of course. Yeah, any other question? Yes, please.
I'm curious about that hydrogel.
Yeah, go ahead.
In that hydrogel, in the graph you showed, I think the hydrogel itself degrade very fast, and the granulars stay at the surface of just like glues. So, how much?
Yeah, I just want to show you guys the last few slides. So that at least I want to repeat them, but at least show you some general idea.
Yeah. Laser to transmit power wirelessly too, right? Just
Absolutely. Unless there's another. Okay.
How many ports take to screw on
Yeah, it probably will work.
Yeah, it's here. I can write it.
相当于pH responsive.
Okay,
really
yeah,
enjoyed
yeah,
your
thank you. Thank you very much. Apologize, just want to show you the last few things. So if someone can turn that on, that would be great.
screen. Yeah.
Project very small.
Oh, no, I don't.
Some glass in here.
Okay, that's great. So I mentioned about the surface modification. It's very brief. Use this modification, you can actually through MD simulation, you already see the difference, right? You can enhance the interaction between the granular particles and the surface of the cell, the membrane. By the way, this is MD simulation done by Peter Crow at USC. And we have tested the adhesion in a range of models from cell culture to organelle model or the real tissue. Won't go into the details. And I mentioned that this can be used for a range of applications. Applications include the treating of ulcerative colitis, and you can form a temporary biophysical barrier and help with the reconstruction of intestinal barrier. And you can also use this for a bioelectronic composite attached to the hard surface. What you can see is that there's an increased recording signals because the granular particle, that interface, fluid adhesion interface, is actually better in this case. You can apply it to the ischemia and reperfusion heart as well, and you can clearly see a difference in terms from this isochrome map in terms of the cardiac propagation, signal propagation. Now, the last one I mentioned earlier, I started with this direction back to 13 years ago when I was still a postdoc. And that time we used a bioelectronic scaffold, engineered tissue scaffold, but there are also bioelectronic components. We have incorporated cardiac cells, the newer tissues, and also vascular tissues. But we use this mostly for drug testing at that point. So throughout the years, we have started intracellular integration and using various methods, biomineralization, or using this phagocytosis process, but we cannot really call them bioelectronics until very recently. So the basic idea is that we have three pillars for constructing bioelectronics. Traditional pillar is bioelectrical. Okay? You only need to consider this part. And in recent years, people started to think about biomechanical. For example, adding some hydrogel component to reduce the mechanical mismatch between electronic part and the tissue part. So our contribution is to add the third pillar, which is the biogenic. We have the biopolymers, living cells. And these living cells can secrete important molecules that can be helpful for wound healing or inflammation treatment. This is how the device looks like. You have the sensor part, you have the stimulation part, but you have the living hydrogel where you have the living cells. This green dots, there are the SE6 living bacteria. And as I said earlier, you can modulate the activity by passing electric current. You can also kill them if you don't want to use this. And we can use the sensor parts to measure like skin temperature and impedance. This can monitor the progression of the inflammation and recovery process. These are just some idea about for future direction. We're excited about precision electronic medicine, where you integrate sensing and modulation component in closed loop. We're excited about non-genetic synthetic biology, especially if you can further reduce the size of the semiconductor components to the range of single molecules or even organelles. Some recent work done by Stefan from UK showed that protocells could be used for biomanufacturing. So we do feel that semiconductor-based components could be incorporated into this as well. Now, I'm also a big fan of the microbial and mammalian interaction, this nexus thing. And we believe that bioelectric signaling or connection may actually help those two words to talk to each other. The last thing is what we call bioelectrical morphogenesis, or morphogenetic engineering, or morphogenesis. So by controlling bioelectric signals in a spatial temporal way, you might control the developmental biology process. Now, the harmony and the beauty that we see from the nature is exactly what we hope to emulate in the synthetic materials and devices. We're scientists, but we are also the keen observers from nature and learning the nature's wisdom and apply this to the engineering devices and tools that can work with biology. So with that, I would like to finish my talk, and I would like to thank my current students and postdocs, former student postdocs, and a lot of collaborators. So Naru Hibino, he's the cardiac surgeon, and Guru, he's the microbiologist. He's the San Diego. So a lot of other people, I won't mention their names. With that, I would like to finish our talk and apologize again for the issue, technical issue today. Yeah, thank you.
All right, that's great. I guess.
Thank you. Thank you. Yeah.
Sums up for finishing the talk. And thank you for staying towards the end. And I guess we still have a few minutes. Let's see whether the audience have additional questions or not.
Yeah, please.
Is that you use the hydrogel as I think the hydrogel itself degrades faster than the granules.
The hydrogel, yes, they are more responsive, right, to temperature or pH. So I wouldn't say degrade, they can just melt. The temperature can melt them. Yeah. And then the granular particle release. It's all controlled by the viscoelasticity of the matrix.
Skin or other absorb the
Yeah, gelatin can be easily absorbed by the body. Yeah. It can easily degrade too.
how long?
How long? Well, I would say on the order of a few hours.
Yeah.
I have a question about the releasing of the granular. So as you mentioned that the hydro will be like melted through the alterating of the pH or the temperature. So how do you achieve the pH changes to like?
Yeah, think about like our GI tract, right? In the stomach, the pH is acidic, right? It's a very acidic environment. But in the intestine, that's a more like basic or a base condition. Yeah, alkaline condition. Yeah. So there's the pH changes. And if you, let me see. So if you look at that curve, when you increase the pH, the viscoelasticity will become more viscous, meaning that it can be released better. So the material in the stomach is still a composite. You have the hydrogel wrapped around, and that can also protect granule from early degradation. But then in the intestine, it can get released, and that granular particle can dock on the tissue surface. That's the inflamed epithelia. Yeah, sure.
Maybe this is the last question.
Okay, I have a quick question about your minimally invasive device. So you said you can apply the membrane. Can you also extract it off of whatever surface that you put it onto?
Yeah, the minimally invasive, the first part of work, or
Like with the, I believe it was a pipe and.
yeah.
So you can obviously apply it
Yeah.
to surfaces. Can you extract them?
Yeah, this is a great question. So at the current state, so silicon membrane is actually biodegradable. Depending on how long you want to use it, it can degrade naturally. And the degradation product is called silicic acid. Every day we eat vegetables, and we actually have silicic acid in our body. It's part of the degradation product from vegetables. Yeah. And it can be cleared by renal clearance.
Thank you.
Yeah, of course.
Oh, yeah, I forgot to check. Anyone, let me check whether there's any questions in the Zoom, okay? But no, I don't see any. Message here.
Oh,
Okay, all right, maybe let's wrap up the seminar. Let's thank our speaker again.
Thank you very much. 不好意思，不好意思。